So this is about latent space learning. Well, I think that the field has as a basis for many downstream operations is that we kind of operate on a robust representation of this high-dimensional, potentially multi-view and for sure noisy data. What do we have? Let me just go through my standard intro slide because I really like stressing the point that this is big, right? You know, we have, for example, droplet-based assays, but now with combinatorial indexing and so on, our data sets are getting huge. They're getting in a range. This has been a method of years some time ago. I'm sure you're all aware. But this is really going into a range where we have hundreds of thousands of cells, where our covariance matrices finally really also in this tens of thousands of transcript dimensions tend to become invertible. So, you know, we're getting into a range where condition numbers of these things get robust, where we could potentially hope to learn regulatory networks. So this thing has been catching my interest early on at the time where we still qPCR and then later sort of getting larger, thinking that, you know, you could really start doing some more machine learning and sort of maybe also push methods on this field. I think this has been very, very, very promising ever since. This is from an old picture on sort of the standard work from single-cell transcriptome analysis. This is from Rick Sandberg quite some time ago. The idea is, for example, you have healthy data, you have pathological situation, these small cells pop up, and then the sketch is kind of clear what you would do. You would do single-cell RNA-seq, you do some clustering of the expression profiles, and then you embed them in low dimensions. Then you compare those two embeddings, and you quickly pick up those disease-associated cells. And that's still kind of what we do in terms of workflow. But for each of these steps, a whole bunch of issues arose and sort of kept us busy, coming from, you know, these clusterings, but then also the mapping. You know, we really asked all these things again. I don't know how many algorithms for clustering around with this specific flavor in single-cell RNA-seq, but also comparing those dimensions, doing dimension reduction, but then comparing those distributions. There's a whole bunch of things to be done. And I guess we're still working on that. So that's why I think this grows the whole set of computational challenges. My lab, together now with
Hi, Fabian. It's great to see you again. What do you mean by the mapping? When you say mapping, what do you have in mind?
Oh, yeah, that's a bit simplified. So I basically stole that. Well, this slide is from Rick's review. So he called it cell type maps, right? I think nowadays you would just call it dimension reduction or visualization. And in his sketch, I think he was thinking about the typical T-SNEs. What I want to sort of put critically in this map is, of course, this visual comparison is not what we would do statistically. Statistically, we need to compare those dimensions, not in 2D, but in high dimensions, obviously, right? But yeah, I mean, as a map, in this picture, it would be really just a visualization. I'll later define what I think what an atlas would be. And I think an atlas is more than just a visualization of this expression profiles, okay?
Yeah, that's great, thank you.
Yeah, so it's a good point. Map it's a bit arbitrary. Map, right? So yeah, I said there's a bunch of tools around that we and others contributed, and I think Peter just gave a very nice review and also critical illumination of still ongoing issues of these so simplistic looking steps. And I encourage you to read it if you haven't done so. But my key point here is sort of going back to the title, that those maps, which is a bit unclear, which often are just, at least in the early times, were used for visualization. And, you know, the people look at the T-SNE saying, hey, this is sort of my data, so this is my atlas. In reality, what we actually mean is, I think, a latent space. This latent space could be then 100 dimension or something, but it's sort of the manifold where all the data is sort of lying on in terms of the true structure and the noise being removed, and typically also integrated across batches or things like that. And I think this is what most of this downstream analysis are being based on, at least for grouping cells, for finding trajectories, and so on. So I want to talk about this idea of latent space learning. Just to put things into context, Luke, a postdoc in the lab, has early on been driving and sort of collecting a database on single-cell RNA-seq tools. And I think it's fun to look at what people are currently working on, and this is sort of what he enabled with that. And he had an early paper 3 years ago, and recently we wrote up now a small text about now being more than 1,000 tools in this data set. Just seeing where trends are going, I think it's interesting to see if we plot sort of the proportion of what tools are in the database, and you'll quickly see that clustering is going to be a big thing, as you saw from this previous claim of the sketch. But I think it's interesting because he has these things, he can sort of really look into abstract and see how they develop. And what you would see is, you know, people are still doing dimensionality reduction and clustering, So this was this mapping idea, visualization also, most of it actually coming popping up in a lot of tools. But over time, but these are sort of not getting as popular as they used to be. But over time, classification started to become a thing, perturbations, but in particular also integration. And I think a lot of things that we think about with respect to latent space learning actually connected to this data integration question. All right, so what I want to say today is that latent space learning, depending on the constraint that you put, I think this constraint is absolutely important. We just speak about one-dimensional latent spaces, which are trajectories, trees of that. If we integrate them across studies, this is atlas learning essentially. If we do this across perturbations, could be disease or drug or something like that, you'd be sort of needing to take these covariates into account. And if you add now multiple omics levels to do multimodal or multi-view learning, as it's called, machine learning, right? In the spatial context, you would sort of need to do something with neighboring cells. So I try to put each of these things in context, and I would ask Maren to present one of her actual benchmarking studies on these things later. Natasha, I do see some chats coming up. You let me know if I should interrupt and say something, right?
Okay, yeah.
Cool, thanks, man. So let me get started with this simple thing. I think most of us have seen this. Just want to point it out again because it kind of also gives us the illustration what this latent space could essentially be. So if you really look for one-dimensional latent space, or if you extract one-dimensional latent space about a high-dimensional one, you sort of sort cells by similarity, right? This has been one of the first sort of big set of tools that have been around, one of the earlier ones, which is, you embed those cells, high-dimensional transcript space. Typically, they don't form a blob, but they might be sort of specific somewhere. You see an example. This was the qPCR, but if you just do normal clustering, it's not so much structure to be seen here. This is work together with Göttgen's lab from Cambridge, UK. In reality, those cells would be lying on this submanifold, and there might be progenitors here. Cells might be rolling down this Wadding's landscape. We want to find this submanifold in the high-dimensional data set. So this is classical challenge of dimension reduction if things were linear, or non-linear case manifold learning, right? And one of the simplest ways to approximate described manifolds, I would say nowadays, even if one of the most popular, is to use k-nearest neighbor graphs. If we have such k-nearest neighbor graphs, so just doing Euclidean distance high-dimensional space, maybe doing a bit of normalization, but it's not as simple as that. But just connecting nearby ones and filling them up, and then for example, in our case here, let many random walks run down these k-nearest neighbor graphs. We can measure distances. We can build something that's actually close to geodesic distances. And there, I think many of these ideas have been then building upon this. So I think since the first tools such as Monocle, which was doing a min-span tree on that graph, or Wanderlust from Dana Pair, which was sort of sampling random walks, and then our later diffusion pseudotime, which was doing this sort of the sum of all random walks, also with branching. So these things have been around, and I think nowadays we can rather well sort cells by similarity of their profiles. And if you do this, for example, for this data set, we very nicely see turning on and off of particular programs. And so this has been really popular. Many tools have been proposed. And I think this is something that we see can becoming more and more popular in our field. Then also reviews of those tools, but also benchmarks of those tools will come up. This is a very careful benchmark, very, very, very detailed, very nice done. And you can sort of see which of these things actually work robust. Obviously, it always depends on the question, the data set you put in, and you'll see later from now that there's a bunch of challenges involved as well. So the summary of this idea essentially is, we have with this thing, we can use graph-based, we can also use neural network methods, which I'll talk about later. But essentially, we learn a one-dimensional latent space, which might be curved, potentially branching. Simple setting. I just threw in one slide on the dynamics because I think it's such a fun thing, even though I won't be diving more into that. Many of us obviously have heard about RNA velocity. And I think this learning about trajectories is very tightly coupled to learning how cells transition into a new state, let's say during development or during response. So in a sense, interrogating this dynamical system here. So I'm really, if someone wants to discuss on this, I'm very happy to hear your feedback. But I think this is really essentially, I mean, obviously I write it up as a deterministic thing. There might be a lot of stochastics happening and all kinds of things. So this is hard. But essentially, if we know how a gene expression state of a cell would transition into, if we fix one, could transition into a new one, how it would change, we'll be able to explain all these branching, this transition events and so on. And in a sense, this function here you could derive or potentially even call a gene regulatory net, right? One of these very simple approximations of this thing, now not fitting the derivative, but actually putting it together, has been proposed by Peter Kachenko and Stinner in the RNA velocity approach. So in this case, they've been actually observing that in RNA-seq data, across actually a bunch of techniques, you find both not only spliced, but also unspliced reads. So what you would see is that, for example, those reads would be processed and give me a sec, my daughter. Paul, ich mache gerade einen Vortrag. Du musst mich jetzt, du kannst nicht zurückschneiden. Sorry for that. Home office. So we see those, we see these splicing events. We can put this type of sequence, we can put this in a model. And if we do this per gene, we can actually put, for example, plot unspliced versus spliced reads. You would see in these regions, when you have more spliced and unspliced, you would have an up, you would have a down regulation, and you can fit these things. In our case, we could extend this with this Civilo model in the fully dynamic model. But essentially, you can approximate these networks. So I think actually using these type of approaches, not only for learning trajectories, so where cells will go in the next step, potentially also what's driving this would be quite exciting. And I'll sort of finish with a conclusion in this sort of more causal direction at the end as well. But at the moment, our latent spaces are really just mappings without maybe initially understanding that too much. All right. There's a perspective on RNA velocity where we think things could be going that we wrote together with Peter's lab. If someone's interested in this. All right, let me continue with
I have a quick question. We can have a more detailed discussion maybe afterwards, for the interest of time. But I just want to know that, you mentioned that you can fit sort of this, dynamical equation of data. How would we then be able to sort of decouple or disentangle, the cell autonomous effects from, the non-autonomous effects due to cell-cell interaction potentially? Because however experiment we design, I guess it's very difficult to control the effect of other cells.
Absolutely, this is a super important point. So clearly, this is much too simplistic what I wrote down here. In practice, and this is sort of what people in old-time systems biology, how many of those around, have been always capturing with sort of additional term external or something like that, right? So for sure, this would be sort of a self-autonomous type of thing, and you should write it down in a stochastic fashion. I've been really being simplistic here, but yes. So there should be also another term that could be cell-cell communication, so really just sort of neighboring cells, but also of course perturbation, or sort of external turning on and off of things, changing with media, whatever. So yeah, these things are often obviously not happening in a vacuum. Yes, important point. And of course, if you know those functions, you could potentially also put them into how those would be influencing gene expression. This is sort of really a high-level view, putting down those Fs in practice, from Hill kinetics to just really simple linear models. There's so much things around, but I think there's some successes of this are starting in our field. I'm sure many of you have seen Cell Oracle. In this case, Sam really just uses a linear model for that, and she can sort of extrapolate a bit in the future. And I think also Peter in his paper has been sort of pushing things into the next stage. So I think these ideas will become open.
Thank you.
Right. So this was about the one-dimensional case, about essentially transitioning from one cell state into a new one. Let's take a step back and sort of think what a larger dimension latent space learning would mean. For this, I think it's essentially about trying to take covariates away from data, or sort of really learning the overall variation and then describing this somewhat, potentially visualizing. This was the summer what we already discussed, or then sort of integrating with other data, essentially building atlases. And since this has been originating at the broad, everyone has seen this logo and the Human Cell Atlas. There's a sort of something downstream equivalent in happening in Europe called the Life-Themis HubMap and so on. But the idea is we want to build this periodic system of elements equivalent of cell types. So what would be such a cell atlas? I check out what we wrote in the Human Cell Atlas webpage, and I think that might be just taken from the white paper. At its core, a cell atlas would be a collection of cellular reference maps. So just going back to what we earlier discussed. So yeah, here's the map notion coming in again, and this map should be a characterization of thousands of cell types in the body where they're found. So in this very simplistic notation, cell atlas would allow us to annotate cells and where they're found, sort of put in spatial context. If you want to be a bit more mathematical, or at least a bit more explicit about that, I guess what we really look for is, I sketched again a two-dimensional thing. This should be a latent space, okay? So not a team, but if we have a single study, we'd have cell type, we'd have a bunch of cells where we annotate different cell types, and then we would be happy with this particular readout. But in reality, an atlas is not just a single data set, right? It might be multiple batches, but in particular, it would be multiple studies. So an atlas should be robust under generalization. So it should be the usual problem that we can see sort of if we compare RNA-seq across labs, even in bulk, we saw strong lab effects. And this is, I think, the case in single cell even more so. If you do multiple of these studies, the cell types would often be somewhat in proximity, but a strong dominant axis of variation would be a study, a batch, or something like that. So a task to now resolve and essentially analyze this consistently would be now to do data integration. And this is, I think, one of those challenges that have been around the past 2 or 3 years, and a lot of people have been thinking about how to do this. Essentially, now superimposing these colors, these would be the cell types, superimposing them onto one or other, and then being sort of somewhat consistent across the different studies. So removing essentially this step, variation in this manifold, orthogonal to the biological variation, but removing the study.
Hi, Fabian. I'm Hirak, Peter's Karchenko student. So I had a question about this, a high-level question. I think I asked you before this question. So what does it really mean in terms of distance in the latent spaces? I think you answered me that it depends on the data type, on the latent space we are looking at. Has there been any development since I asked probably 1 year ago? And do you have like more insight on how we interpret really these distances? Do they really mean anything biologically?
So you're asking about those specific distances of distance between study versus distance between cell type, or in general how we calculate distance on those?
No, specifically I'm asking in the context of the atlas situation, because for a particular sample we might understand that this would be clustering of cell types or probably what state they are in the landscape. But in terms of atlas, it's much more complex, I suppose.
Yeah, this is something where we've been actually having a small subgroup of the HCA analysis working group, where we've been thinking, hey, how could we properly describe how to first build, but then also interrogate these atlases. So I think you have a valid point there. So surely, right? I mean, if we have been able to do this type of integration successfully, then again, we would potentially be able to interpret this. Often this is something nonlinear happening, so it's not such an easy to understand map. But often we would be then able to understand that, small variation in here would be then just variation within cell type. And that could be, for example, cellular state. There might be also connections between these, like tubes, something like that. There would be trajectories. And the big variation across this would be then dominated by cell type. But in practice, I think it hasn't been fully discussed. So this is what I put down here. So I would say as a summary, what is a cell atlas with one data set? I think it's kind of annotation. With multiple data sets, it would be this integration. And here what I illustrated would be this joint latent space. But I explicitly put a question mark there, maybe a little bit what you're hinting at. I think there's no final answer whether the best solution for, let's say, shuffling out a full human cell atlas would be us being able to essentially smush away. This was a term copyright Dana Pierre, she called it smushing, or away biological variation across, not a study variation and keeping the cell type variation, because obviously there might be the big danger that we might be removing important variation. So there's also sort of an opposing idea of being able to map between each of those data sets, but not doing this jointly. And I'll discuss it a bit further. So you just keep the thought. I think it's important.
Thank you.
All right. So last slide on that. There's been nice review from John and Ollie on data integration single-cell genomics, and they've been sort of putting up different schemes. And the one we're talking about here would be different labs. so we call it horizontal integration. You have a bunch of features, okay, it's RNA-seq, measure cells, you need to smush them together. And there will be orthogonal other ones. Let's say if you have multi-omics that you would want to integrate them vertically. We'll be speaking about this multi-omics thing later. But now I would hand over to Maren to illustrate that we've been really looking at a bunch of these integration methods and try to deal with it.
Thank you, Fabian, for the nice handover. So I would like to illustrate the problems that we have with integrating data on the lung cell atlas. So what you see here as an embedding is several data sets that we have, and we colored here by donor. On the right-hand side, you see the cell type annotation, and you see, let's focus on the macrophages, which are just encircled here, and they're kind of all over the place. So when we compare the left and the right image, we see that these macrophages do not align with the cell type, but they rather align by donor. So we would like to get rid of this donor effect, and we have lots and lots of different methods available. So yeah, we just named a few here, and I think this is already kind of overwhelming if I have that many methods at disposal. So what should I do? So the naive approach would be, just pick some and try which ones the best or how which appear usable. And here we have selected SCGen, Harmony, and Desk. And like by looking at the low-dimensional embeddings, we would say maybe we go forward with SCGen and Harmony. And here you see again the cell type labels. And when we now again look for our light blue cells here, more in the center, and we compare how they are embedded. So we see one goal we achieved is that the donors now nicely overlap, but we also see differences in the embedding with other cell types. So whereas in SCGen we have a nice distinct cluster of macrophages, we see that in the Harmony embedding we have a connection towards neutrophils, which might not be like the best way to embed this. So maybe this is already some kind of an over-integration, and looking at lots and lots of these low-dimensional embedding is not really a good way to quantify. So we sought out to evaluate how data integration should work. So again, we made a selection of almost 20 methods, and we start out defining that we have the batches. Batches are known in our data. And if we want to integrate all these different batches, the lower picture would now show us a good batch integration. So all the cells coming from different batches, the dots overlap. However, if we add the cell type information, the picture is largely different. So when we want to have a good integration that preserves our biological information, we would need the cell types to cluster, whereas different cell types should separate. So these are the two different metrics that we are now trying to establish. One, defining a good integration in terms of batch removal, whereas on the other hand, we need a good conservation of the biological variants. Here we have distinct cell types. Usually the annotation also only handles distinct cell types, but what is more, we could also look at continuous phenotypes. So what about developmental processes? It could be as simple as hematopoiesis. So we have a continuum, and we want to preserve that continuum of it once we integrate different data sets. Or we would like to keep the gene level variation as here depicted in highly variable genes. Okay. So let me present you our benchmarking setup. We now created 13 different integration tasks covering RNA-seq and ATAC-seq, and different pre-processing methods. So whether we selected highly variable genes or whether we scaled the data. And then we, let's say, packed all these different integration tasks into these 16 different integration methods. So, and then we get an embedding or a graph or corrected features. And that's also one of the caveats that we have with this method. They all output a different type of they create different types of output. So it's not as straightforward to compare an embedding to a feature to an integrated graph. So what we then did was to set up a whole battery of different metrics, each measuring different things, so that we could average out certain effects on certain metrics. So for example, Combat maybe optimizes for PCA integration, DBCAN and optimizes for like graph integration. So certain metrics would inherently score better for certain metrics, and we would like to average out these effects. So we created 14 different methods in total, and we also evaluated how well these methods scale. So whether I could apply a certain method to 1 million cells on app, and how well they're actually usable. So going from the books to computer, how easy is that provided? Okay, when we now go for the next slide, this is one of the examples how these outputs will look like. And we have here in this example a top scorer, which is harmony. We see that the output is an embedding. And then the next are the features. So we selected highly variable genes. We have unscaled data, and you see here the overall score, and the scores in bluish colors are the batch correction scores, and the pinkish colors are the bioconservation scores. So in total, we have these different methods, and then could create a ranking for each of our scenarios.
A quick question. This is Luca. I'm a machine learning scientist working with Mertash.
Hi.
It's a little bit unclear, at least I didn't understood if you have a ground truth for what you are trying to achieve or not.
Yes, excellent question. So we annotated all these data sets manually. So we had, from these 13 different integration tasks, we annotated all of them per sample, and had also the integration, the annotation backed by, whether this is actually checking out whether this is biologically meaningful. So we have, for example, annotated endocrine cells in the pancreas. When you look into data integration, integrating pancreas data is one of the standard examples. So that's why we thought it's well established. We have to look at those data. We also looked at mouse brain studies. So that was our large scale example, 1 million cells from 5 different studies, and we annotated them based on the publications, as well as harmonize the labels so that we have the same labels in all of the studies. Yeah, I hope that.
Thank you.
Thanks, answers your question. So we have the scores, we have the metrics, we have the outputs. We could create an overall ranking, and we can compare now how well all of the methods are doing in terms of batch correction, and how well they preserve the biological effects, information in our data. And what you see here now is that each of the method has to balance these two things. So if you are good at batch correction, you're potentially not so good in conserving biological information. So, and the best method should now be located in the top right part of this part. So being good in batch correction, as well as being good in bioconservation. So in here, what you can see now is our top scorer are maybe, go for that side. So to make it a bit easier for you to see what our top scorers are now in terms of an overall score. What you see here now is, for example, SEI, which is consistently among the top performers overall types of tasks, whereas, for example, Harmony did very good and the pancreas task was talking about, as well as Asura, but these tasks tend to be, and this task, but the these methods tend to be lower or less performance in the more complex tasks, like we, for example, included an integration across species with immune human mouse. So that was particularly challenging. Yeah.
Hi, Maren. What is simulations here?
Simulations are distinct clusters of cells. So we have two simulation settings. So the first simulation was essentially, let's say, distinct cell types, one type of batch effect. And here you see that, for example, scANVI and scANORAMA, or also DESC.
Is it running? Is it like running splatter with some ZINF model or something like that, or it's? I
think it's something like that, right? But
yeah, it was splatter.
But you did not use, right?
Yeah.
But
yeah,
you
did not. Splatter.
But no, you did not use this simulation for the overall ranking. I think the overall ranking was based only on the real data. Is that correct?
Yes. Yes, that's
okay.
Because simulations always be debated, right?
Yeah. Sorry. Yeah. Simulation 2 is having a nested batch effect, so that we have different levels of batch effects. And some methods did very well for this, and others, didn't. So we tried to cover as many different settings that you could have as possible. Okay, what you see also in this overall ranking is, first of all, the methods that have a star are the methods that also got a label. So scANVI and scGen use the label information, so the annotation of each of these data sets to integrate. So that's kind of cheating, if you ask me, because if tell the methods which cell type is or which cell is which cell type, it has to perform better than a method that doesn't know, like all the others that don't have a star. So overall, what we can take home from this huge benchmark is that Seurat and Harmony perform particularly well in these less complex tasks, be it the simulations, be it the pancreas integration, whereas scANORAMA, SCVI from near Joseph Lab, scGen from developed by Mo, and scANVI also from near Joseph Lab, they perform well across different integration tasks. So for me, as a data analyst, looking at various different data sets, my first choices would be SCVI if I have enough data, and scANORAMA like the first try. What I would also like to stress here is that pre-processing matters. So that's why we also listed it here. For example, if you see scANORAMA is listed twice in our ranking. So third place is scaled HVG selection and an embedding, whereas on 8th place we have both scaled and HVG selection, but the corrected feature space. So that's, thank you, Fabian. This is something that you also have to take care of when the method can perform worse if the data is not properly pre-processed. We have written up all these methods and data integration tasks and metrics in our SCIB pipeline that can be run as a snake-make tool.
Cool. Thanks so much, Maren, for the summary. So I really wanted, we appreciate Maren, really wanted this to be included because I think we had this so many tools around, and I think benchmarking is one of the next steps. Also, I wanted to highlight, because actually today we started our lab internal hackathon, and this is something actually a lot of people can contribute. So this was one of these results of a hackathon we've been doing, I think 2 years ago, and then sort of 1 year ago, and concatenating. In any case, I think it's a fun thing also to run benchmarks, and sort of to maybe motivate more people to participate in the benchmark. I think there was an MIA talk just recently from John Bloom and others. Particularly Dan Burkhardt and Malte Lücken have been setting up this open problem single cell analysis, where we actually also focus now on multimodal data integration. So if you're interested in having your own tool there, comparing, writing a bunch of metrics and so on, please participate at this competition. So this is not going to be this task, it's going to be this task. potentially also a little bit this one because it's about across multiple labs. These would be the different types of tasks to look into. There was a talk about it, so I won't go into detail. I just continue with the second part of this presentation. We will be using now something so very scalable for latent space learning. You saw already that SCVI, as generally related things, have been performing quite well, particularly if datasets get larger. So I think it will be hard in the future to beat the flexibility of neural networks. So let's maybe think a little bit how we can sort of do these in our setting. I think the simplest architecture, it's always unsupervised, in most cases at least initially unsupervised in our setting, is going to be based on this autoencoder. The idea being that, you know, you take your gene by cell matrix, you squeeze it down, you blow it up again, and then you make the output as similar as possible to the input. If everything was linear, you know, this would be just learning a PCA. If the loss was just R² type of loss. And because we have component-wise nonlinearity, we sort of have a nonlinear generalization of PCA if you want. And, you know, because this sort of cleans up things, you could, for example, use it for reconstruction. And this was sort of one of these early approaches that we've been also looking into. But the most interesting part is really that this bottleneck layer actually could lead to some of these interpretable things. This would be the latent space, really. So doing stuff then in this bottleneck, I think, is where a lot of interesting directions will be happening. Let me show you one of these early examples. This is from so there's a whole bunch of tools around. This is what we call deep count autoencoder. I think our DCA together with NIR's SCVI have been the two first things we went actually back to back on bioRxiv at the time, and I think it was really fun, more or less back to back. It was really fun seeing how this has been developing. In our case, not even a variation version. This has this beautiful noise model included. We essentially just extended the noise model here in the autoencoder, and we're able to then do denoising. We showed this on a bunch of simulations. I won't go into detail now. And obviously, this thing is much more scalable than any other type of approaches, because, you know, it's a neural network, so it just goes to much larger data set. What my main finding in this was, and this was sort of motivating a lot of downstream things for us, is that this bottleneck layer here actually shows interesting results. I mentioned this before, but in this case, it's an explicit PBMC data set. Each of those colors would be different cell type. And we've been driving this bottleneck, usually it's much higher dimension. We've been driving to 2 dimensions. And it really learned something that looks like a T-SNE map. It really aggregates cells by the similarity, by the cell type, essentially. So this thing kind of needs to do something important. This kind of needs to encode something about the regulatory interactions. At least you really have an explicit map of that thing, of that high dimensional variation, and sort of now putting constraints in there could potentially teach a lot of things. And this is the idea. This is from a recent review from NIR's lab in MSP, where he has been now speaking not about normal neural networks, but those generative version where you actually then start sampling in the latent space. This is more flexible in the sense that you get some kind of uncertainty estimate, and you can also generate samples. But the idea being that whatever data you observe, he has been sort of putting up in a more general setting that there could be also the DNA sequences or encodings. We're talking about those sample by gene matrices at the moment. And with a generative model, you would be learning somehow the true unknown distribution of the data with this generative model. And in the end, this is what our atlases are, right? In the end, we don't really care about particular single cell, but because it's going to be measured to noisy anyway, it's going to be destroyed, but about the distribution of those cells and the groups of those that would be, for example, cell types, transitions, trajectories, and so on. So I think it's a very nice general concept how to approach these, and the flexibility of those deep models, of course, nice. They're not as deep as you want them. Just as a side remark, they're actually not. There's no convolution structure, at least initially applied. So it's really just tabular type of data. So I think there's quite strong potential for improvement there. But, you know, in this review, NIR has been outlining a bunch of applications where things could be going. And one thing would be, for example, if you have no high dimensional observation, you could actually embed this in low dimensional space, and then you can use this for clustering annotation. So this is essentially the idea that I've been trying to sort of put into this notion of latent space learning. Most of those analyses happen on this space. That's why, you know, you can, for example, also use this for supervision and so on. Or in this high dimensional space, you could potentially look for specific interesting regions, sample from that, or potentially even go in between, so interpolate. And this out of sample thing, this is something where a lot of ideas have been going into, learning about perturbations, doing something such as optimal transport, potentially across time point and so on. So there's, I think, a lot of interesting directions happening in this direction. Now I need to ask you, Martash, we sort of should I stop at 45, or should I continue and sort of discuss with questions between.
Oh, no, please continue. Yeah. Continue for maybe another 10 minutes.
Cool. So just thank you so much. So just briefly saying something about the permutation, this is something that we very often do, because now we've been sort of copying that idea, which has been so much longer around the computer vision, that we want to use generative modeling there. How could we apply this in our setting? So I think it's always good to look over there. They've been doing, for example, this image-based, these face database thing. It's this really nice sample from a latent space, and then do some type of arithmetic in a latent space. You take men with glasses, subtract men without glasses, add women without glasses, and you sort of generate images of women with glasses. I thought that this was really fascinating. There's a whole bunch of, and it's actually really nice. If you do this type of arithmetic in latent space, it actually works. If you do it in pixel space, it does not. It means that the latent space sort of needs to learn something about the relative notion of eyes to nose or something like that. And I think a lot of sort of more complex things, such as these deep fakes, are based on this idea. So this is really going out of sample, and you can even go in between, interpolate. So in our case, potentially maybe learn transitions between cells, maybe even think about reprogramming and so on. So I think there's a lot of ideas that could be held. So we've been thinking, if you have a cell, maybe you have a perturbation, such as adding an overall general drug or doing something simpler. But if you then sort of put those into context, you might maybe learn, if you have an observed perturbation in one cell type, you might be able to predict it in a new cell. So this research aimed prediction of perturbation effects, potentially interpolating in new cell types, potentially in learning a full perturbation atlas, just based on a few examples, could be possible with this type of generative out of distribution type of ideas. Obviously, it's going to be noisy, it's going to be all kinds of issues with that. But I think this as an outlook towards this typically infinite dimensional space of experimental perturbations could be quite exciting for experimental design. We had a few ideas for that, SEGen. I won't tell you about this now. I just want to highlight that Mo will be presenting this and extensions in the next talk. And I just want to sort of highlight one more review, that I think with this idea of using some high-dimensional phenotyping, namely those things scRNA-seq, for, let's say, readouts of cell and how they behave under drugs, could be a quite interesting way to model also more complex behaviors. So this is from a review we recently put out. This already showed that, if it's this hourglass-shaped type of encoders, you could potentially combine this with phenotypic information. You could include then target mechanisms, something that's not yet happening, but you could also see how perturbations potentially interact. Again, Mo will say a bit more towards that, and potentially even include something about chemical properties. So I think linking now, once now we have a good, robust model for this observation of expression, so this intermediate phenotype type of thing, which is sufficiently high dimension to describe a lot of variation, we could now add other types of mechanisms into that representation. So the end is always about representation learning. I think doing multi-representation learning will be one of its next steps. And why is that important? I mentioned this already. We can only do so many experiments in this whole space of potential experiments. If you can even just extrapolate a bit, this was this dotted line from near, which you call out of distribution, which in parts we can actually do explicitly, as you might see from Mo. This, I think, is going to be one of those interesting new directions to really use those atlases for reference mapping. So in these remaining 6.5 minutes, I want to show two more points. The one point I want to show is that if we now want to use those reference atlases, those latent spaces, we actually have to do some work. This has been put forward already by, I don't know if you saw that this was this 11 grand challenges paper in that many of us contributed to at a very nice workshop in, I think it was in Belgium. And one of those challenges was actually mapping single cells to these references. Because, we have those references, but we don't, what we very often do is we annotate each of these new data sets again. But in practice, we just want to map onto this reference. We want to use this annotated reference. There's a whole bunch of challenges that are involved in doing so. So to do this robustly, you essentially need to take your reference and anchor some of your query on that reference. This is from a paper from Raul Satija, who has been putting forward his CCA-based reference assembly. This was the third method that Maren also just benchmarked. There's been a bunch of other ideas for doing so. What we've been doing in this paper, and this was also a work by Mo some time ago, we've been taking public reference data sets and then train one of these autoencoder models that I showed you before. This could be quite a bunch of them. In practice, you just take a DCA or use a conditional one of that. So this would be, for example, conditional ratio autoencoder, such as implemented from STVI. or TRVE, and then you have the reference label included here. And the idea is now that we don't store the studies in the end. We just store parts of the data, namely just the model. And then we can actually do transfer learning across states. So the transfer learning idea, I think, is the key part here. We don't want to just do this again for each new study de novo. We only want to keep that part here, and thereby we can add new study. But how do we do this adding of the new study? Well, we need to extend this reference label that I embedded here. And we show that in many cases, it suffices just to add one more node to this network and connect it and retrain on this part. That's why we call it architectural surgery. And these things that we add, we call adapters, which you could do for multiple ones. So this has been something that has been actually working out quite well. I won't go into detail of this, but it's, I think, a useful concept to transfer some of these ideas that we have into the future. And just to push one more point here about sharing models instead of data, I think generating such a single cell atlas and sort of viewing it as actually a data manifold is a useful concept for interpretation. If you have the human cell atlas here, there might be different areas that might reflect maybe one organ, another organ. There might be some overlap, which could be something like immune cells or so. Or maybe it could be just like two different studies that won't fully overlap. But if we write this up as, we know from maps, where we know how to map in between, then we would be able to essentially describe this complex object of a sphere also in these flat objects that we have. What does it mean in practice? Well, this type of complex embedding thing here that we usually do by hand, it could be reflected by one of these encoders. And replacing now these encoders by a pre-learned encoder, and this is what we do in our tool, which we call Sphere, we actually store a whole bunch of these models across a whole bunch of organs and also organisms. These pre-learned embeddings is something that I think where we can start normalizing a few things. In the future, I hope that many of these things will be actually going from HDA. At the moment, it's sort of a handmade, jointly annotated data set, a resource where we just have a bunch of data loaders for distributed data sets. More than 150 data sets included with these data loaders, more than 8 million cells, and has been very useful for us in the lab to use this.
Fabian, do you have any comments on how to think about the latent dimension in this perspective? I know that's sort of a thorny issue, but I mean, I feel maybe we often make it too low when we focus on visualization, and maybe for trajectory learning, one dimension makes sense, but maybe much more generally, it could be higher.
It's quite interesting. I mean, in these latent spaces in practice, what you would do, you would just treat latent dimension as a hyperparameter and optimize for that, right? And often complexity is not so high, at least initially, if you do it on too many data sets, that it's often fine to have 10, 20, or maybe 50 dimensions in this latent space. Practice, it might be recommendable to use a sort of a little bit higher, and it might be a bit costlier to keep that sort of going during the time, but it's actually useful if there's new directions of variation coming up. So in this architectural surgery example that I showed you, this network is getting increasingly more complex, and there might be new cell types popping up. So it might be good to give the autoencoder one more dimension where the autoencoder can sort of put this thing here.
Yeah, makes sense. But
yeah, it's very heuristic. Sorry for that.
So Fabian, is this going to be used like ResNet or something we use in computer vision, like already trained bank of models, and then that could be just blended?
Yeah, that was our hope. Thanks for putting up. I mean, essentially, I think what we're kind of lacking is this ImageNet, right? Or actually not the ImageNet, which would be just the data set, but as you rightfully said, there's some pre-trained thing. In this case, we have not done a joint training across all the different organs, because that's still hard, even just getting a synonymous annotation. And there's a few papers that start doing this, but I'm very careful with that, because in the community we're still discussing whether we can be even jointly embedding across a single organ, let alone across others. But yes, the hope is that you can use, particularly if you want to sort of, it's maybe you wanted to make a clinical workflow. We cannot do this time and again, and we're getting fast because tools are getting better, but I think at some point, yeah, we need to use pre-learned embeddings. It's like in genomics, we don't want to reassemble the genome every time we want to map onto something. Yeah, this is open. This is all open source. Obviously, I invite you to put your own data sets in. For me, just out of pure selfishness, for the lab, it's very useful just have all our data in there, because then we actually refine our old annotated data sets. So even from that point of view, just for data organization, it's quite useful.
Fabian,
right.
Like as a follow-up, like sort of in the same spirit type question, So I guess here you're proposing the idea of sort of disseminating pre-trained models, right? But there's more like sort of other sort of school of thought, the more traditional in biology that says that we should always disseminate like interpretable stuff, like transcription factors and their motifs, gene expression modules, things that Okay, so what are your thoughts on that?
That's tough. So what we've been trying to do time and again, I think there's been a bunch of papers out, is to actually interpret this thing, right? So this map, I mean, essentially, we know what the input is. There's different genes. There have been a few papers that actually sort of try to go gene ontology categories in there. And sometimes it works. What we've been trying to do is then see what type of layers we learned and potentially add something such as latent space disentanglement, very popular in computer, somewhat popular in computer vision to sort of make sense out of some of those latent space dimensions. Actually, I have here a comparison where we actually try also different types of priors in the single cell VAEs to see what type of additional information we can force. This is an ICNL workshop paper, but I'm not 100% yet sold how to do this robustly. So Metash, yeah, good point. How to share something interpretable, I don't have any idea how to really do this. And I don't think it's happening in computer vision. Nobody interprets the ResNet as much. I mean, you can backpropagate and so on with T-Glyph, whatever, and these things happen on our field, but there's additional steps to be taken.
We can have a longer discussion about this. Yeah, thanks.
Yeah, oh yeah. Thanks so much. So let me conclude. I had something on multi-omics, which I will skip, but I just want to sort of open up super briefly, and I really just showed 2 slides on that. That of course there's more extensions coming. We had multi-omics, mentioned it briefly, but also spatially resolved transcriptomics is a very big thing. There's a lot of essays now coming around where you can actually really observe cells in their local neighborhood, potentially smushed, sort of averaged over a few ones if you take Visium, but these things are getting better and better. There's a whole bunch of fun ideas to do so. We also have a tool for analyzing, which we call SquidPy, which is sort of a ScanPy extension. I won't show that, but I want to make a point going back to the latent spaces. So people have been thinking how to do integration spatial omics. Again, Ollie and John had put up some of those ideas of essentially using maybe Gaussian process regression to see what's spatially dependent and what not. But I think in space, it's really harder. We don't even know how to annotate it consistently, let alone how to really see the equivalent of a cluster or a cell type. In single cell RNA-seq, this disassociated setting, we have this very nice concept, a little bit maybe what you go for, Matash, right? In the end, very often we show UMAPs with annotated regions, which are sort of things that we can imagine. Very, very much harder how to do it in spatial context. I think it has to be something like a local nearest neighbor program, something like a niche that we need to share. So what's the equivalent of cell type in spatial setting? What's the niche there? I think it's really up for discussion. My hypothesis would be that in terms of representation learning, it's not based on a pixel, but cells are the natural unit, how things are touching each other, connecting. So I think doing graph representation learning for this setting is, I think, a very cool idea and potentially useful. Dr. Sidney, actually also from MIT, I think, and also associated to Broad, has been putting this forward again and again. I think super exciting ideas from that. And we've been putting this forward in a recent current opinions that you can actually apply this now for single cell genomics as well. In a bunch of different settings. I won't go into details of this now. I just want to finish with this one thought here, that there's different methods for graph embeddings, node embeddings, graph neural networks, different versions of GCNs based on a bunch of clever tricks. In the next talk, David will be speaking about one particular way how to actually take the spatial context into account. With this, and I mentioned this extension again. With this, let me finish. Sorry for being a bit slow here. I spoke about pseudo-temporal audience 1D building, Atlas as latent space learning in general setting, and autoencoders being one of those tools. Mentioned our model, Sue, and Maren showed the integration part, and spatial learning will be, I think, a fun thing to have.
I'll talk about mostly perturbation modeling for single-cell genomics. I think with the advent of multi-omic technologies, now we can measure many aspects of the cell at single-cell level, and some of them at the same time, basically simultaneously measuring multiple things in a cell. So we would have a holistic view of a cell. And then basically the ultimate goal, and I guess going back to what Fabian discussed, is to build a multimodal reference atlas. Basically, instead of just using one modality, we would like to have multiple modalities. And then we would, after like constructing or like concatenating all these modalities together, we would like to use the integration algorithms that Myron presented, but this time on the multimodal level, to build a multimodal reference atlas. And then basically, so that's the first computational problem, multimodal data integration, and the multimodal basically batch correction. And and then you want to use this like multimodal reference atlases and annotate, and finally, most importantly, basically use these reference atlases. So usability also is another computational problem. And once we have this reference atlas, we can actually query and then answer a lot of questions, and like what are the gene programs that are disturbed, like which cells are impacted by disease, or what is the effect of certain perturbation. So in next few slides, I'll talk about one example of how we can use such reference atlases to analyze perturbation datasets. And then later on, I'll go and talk about how we can basically complete these reference atlases on perturbational data. So Fabian already presented this SDR's idea that we had, and then that's basically using neural networks to build a reference atlas, and more importantly, to leverage those reference atlases to analyze query data sets, to facilitate usage of these reference atlases. So we run integration methods using neural networks, and then we can basically share the weights of those neural networks, and then efficiently integrate novel data sets. But then what is important is like most of these like integration methods, they're so naive, you know, they can remove also biological variations, and those biological variations could be also perturbations. Yeah, so in our ideal case, we would like to have a healthy reference atlas and then project a novel disease data set or like disease control, and then find out the effect of perturbation. So I'll show you one example of like how such examples are possible with our scRNA-seq methods to analyze perturbation data. So in this case, we build a multi-tissue reference atlas, healthy reference atlas, and then we wanted to query this reference atlas of immune cells with a COVID data set from BALF. So we had bone marrow cells, we have lung cells, and we have PBMCs as healthy reference. And then we projected data from 8 different patients, and basically control and then severe and moderate patients. And then we wanted to find out what are the cell types or cell states that actually can arise after COVID. So basically perturbation states, and then what states actually did not affect. So basically we're not affected by disease. Yeah. So here, what you can see here is like the UMAP of overall reference atlas. So the healthy reference atlas integrated with the query. And we basically went and then zoomed in into part of this reference atlas to see which parts of the query data set integrated into the reference and which part did not. So the big difference is like also the most of the cells were from, because they came from lung, were from basically were macrophages. And then we zoomed in wide is like cluster of macrophages. There's like strong difference between control cells and then moderate and severe. And obviously this was done by scRNA-seq, so basically query mapping using scRNA-seq. And then we basically, we could resolve the differences between different macrophage subpopulations and show that the algorithm actually preserved the perturbation effects, basically heterogeneity between moderate and severe patients. And then you can identify that the control cells that from the query that map to the control cells in the reference, they were actually tissue resident alveolar macrophages, whereas this like not integrated part, which was novel, unseen state in the reference, correctly separated from the rest, and they were basically monocyte drive macrophage drives monocytes. So and then we showed basically using different markers that why integration algorithm worked well and why it actually preserved the perturbation. And then we also similarly showed this kind of analysis for CD8 T cells, where we showed that why COVID cells did not integrate, because they were actually from COVID patients, and then the healthy reference, and they were actually activated in cytotoxic CD8 T cells, and the reference atlases were basically naive CD8 T cells, and then data didn't integrate. So this is one approach of analyzing perturbations, but this is more integrative. So you have a reference, and then you project data, and then you want to see what are the differences. And now, as Fabian mentioned, like the perturbation data sets are coming along, and then we have this large perturbation atlases, and we can do and solve, basically we can query this perturbation atlas and solve multiple problems. So basically, I think the most important problem here, which I go deeper, is basically what would be the effect of a certain perturbation on populations that you haven't seen, or like the perturbation that you haven't measured. And then basically that's the computational problem that we have. Or I mean, here we can put it as like ohmic measurement prediction. So given a cell type in condition A, you want to predict how would this cell type would transform into condition B. So condition B could be treated. So the first basically approach we came up for this for this kind of computational problem was scGen. So as Fabian presented, we took some inspiration from computer vision, and then we encoded cells into a latent space representation, and then we came up with like a delta vector that encodes the transition between the healthy and the disease, or basically control and perturb cells. So basically you can take a control cell, compute this delta vector into latent space, and add this delta vector to the control cells, perturb it, and then decode it back to the gene expression space. And this was like 2 years ago, and this was our first effort for this to solve such problem. And obviously what you want to address is, like in machine learning community, is known as out of distribution prediction, because it's not part of your training data. And scGen is available on GitHub. I'll show you one example to basically motivate. So here was like a PBMC data set with both control and interference stimulated cells. And we left out different cell types in stimulated condition, and then asked the model to predict what would be the interferon stimulated version of that cell type. And here you see you see different cell types: B cells, CD4 T cells, and then C, PS, and RS on the bottom. They means like control, predicted stimulated, and basically real stimulated, and or ground truth. And the genes that you see are the top 10 differentially expressed that are cell type specific response of interferon. Yeah. So actually, what is interesting is to find out, and I mean, if there's a global response after perturbation. Could be interesting, but it's not as interesting as the specific responses. And you can see that like with a simple model like scGen and with like variational autoencoder plus the latent space arithmetic, you can capture and predict cell type specific responses. So the model actually picks up like it should upregulate or downregulate a certain gene in a specific cell type that it has never seen in that specific condition, and not basically upregulate other genes. And here's like a quantitative metric. So, and then you can see that we compare it to a linear model, and a linear model is good in predicting global responses. But the first bin that you see in this like box plot, so bin 0, that shows the prediction result are 2 scores for the real and predicted for the hardest genes, top 50 cell type specific genes. So the response genes, and then the nonlinear methods by a large margin can like outperform linear methods. So we extended our method. The method was not end-to-end, meaning that like it was 2-state. You can train a latent space and then you do this vector arithmetics. So we had an extension. I'm not going to talk about this a lot, but like we came up with an end-to-end version of it, and then we formulated the problem as a distribution matching problem. So you want to match the distribution of your treated cells. You basically want to learn a transformation that like shift your distribution of like control cells to distribution of like treated cells. And then we formulated and used like a maximum mean discrepancy distribution matching measure to solve the problem. And then we showed that like the generalizability of the method on imaging. So basically can take the images like as Fabian showed, and then perturb images. And then we showed that this method basically improves upon scGen specifically for cell type specific responses.
How is it different from general conditional VAEs?
So here it's actually a conditional VAE. But in conditional VAE, what you would output would be like still co-founded by the initial condition. Yeah. Here we put an extra MMD regularization that actually forces the inner, like the first layer of the decoder to map. So basically from control to perturb to basically map these like to learn this transformation, and then keep keep the same gene expression response for the rest of the layers. So, and then we basically showed that like the latent representation that you would learn here, it basically, I mean, with a normal CVE, it's not possible to do it because like the predicted gene expression is still co-founded with the initial condition. Yeah.
Okay.
Okay. So TRB is also available on GitHub, but like for most of the talk, I mean, those methods were like preliminary methods, and then we wanted to actually came up basically have an like interpretable method because the previous method were completely black box, and then also address like combinatorial perturbations, so like genetic knockouts or combinatorial genetic knockouts. And to solve this, I mean, because I mean this would be more important for experimental design, since if you have combinatorial perturbations, then the exploration space can explode, and then you cannot obviously at single cell level measure all those combinations. And so the idea was combine the interpretability of linear models with flexibility of representational learning, and to also have like this kind of like factor model inside the deep learning. And I'll show you why I call it factor model, and then incorporate like metadata as like patient level, type, or continuous time and dose into the learning. So this is the work that I did in my internship at Facebook AI in collaboration with Fabian's lab. So this is a collaboration of Facebook and Helmholtz Center, and then this is the method. So we have this is a nice movie by Facebook PR. So basically shows that if you have a cell, we encode it, and then we have a discriminator that decouples the perturbation effect. So here we have the cell type and then the perturbation effects. So you get to a basal state where none of them exist, and then you learn to actually add them back and then decode into the gene expression space. Yeah, and with this trained, you can actually answer the counterfactual question. So assuming that you have cell A perturbed with 2 drugs, you want to predict that what would happen to cell A if it was basically perturbed with 2 drugs. Yeah, so I'll skip that and I'll show you the exact architecture. So this architecture encodes gene expression, and then since we wanted to explicitly model different parts of the like typical perturbation data experiments that you get for a single cell, there would be like you can have time and dose, so continuous variables, and you can have covariates. Covariates could be cell type, covariates could be patients, or like other discrete metadata in the model. So we basically encode, and then using discriminators and adversarial training, you try to disentangle the covariates and the perturbations from the basal state, and then learn a composition that basically takes the basal state and add back those effects. So this is like a linear regression in the latent space where you have like, you basically have different covariates and you actually learn them back. And then these are like all the covariates that you add back here in the perturbations, they are learnable because we have used learnable embedding for them. And then you can basically perform counterfactual inference and then predict like unseen states. So what you can do with this model is predict novel conditions that were not in the data, because we explicitly model continuous covariates such as dose and time. You can model dose response. And then because we learn these embeddings, basically this perturbation and the covariates are learnable, you can look at them in a continuous space of like similar to word embedding when people do it in NLP. So, and then compare like the similarity between different perturbation, basically similarity of different perturbation on gene expression. And then similarly for covariates. So basically if 2 patients are responding similarly to a perturbation. So I'll show you one example. We applied it on.
Do you mind like going back to one slide?
Sure,
the architecture.
Sure.
Yeah. So quick question about the encoder. So my understanding is that the encoder is taking the gene expression and is trying to encode the basal state, basal latent space. So
exactly.
Yeah. So thinking about it from a Bayesian perspective, this is basically it's a variational posterior that's basically taking your observation and giving you some latent variable here z. Now,
exactly. Yeah.
Wouldn't it be then beneficial to let the encoder know what the perturbation and the covariates are also? Basically, now why do you inject those covariates at the on the decoder side and not the encoder side? My thinking is that if you want the encoder sort of to distill perturbation and covariant information from your observed gene expression, it would be beneficial for the encoder to know that.
I mean, you could look at it in two ways. Yeah, I mean, if you look at the Bayesian approach, like if you model this with a VAE or like a CVAE, you can inject the covariates also concatenated with the input in both encoder and the decoder, and then the model is, since it receives this information both in the encoder and the decoder, so it can learn stuff that are not related to these covariates. And then there's like a neat trick in implementing CVAEs that you can actually remove the condition in the encoder and just add it in the decoder. And it will basically do the same thing, because the model is, so it knows that it receives the conditions after decoder. So it basically removes or forget the information that already can remember at the decoder stage. So this was also like, obviously our method is not CVAE. So because we don't use variational and posteriors and also conditional learning to remove the variance of like these covariates, because we use adversarial training. And then for adversarials, it's like, if you have those things at the encoder, then so basically you're telling the model like, okay, this information exists. And then it will be like confusing for the adversarial training, because the model can basically remember that and then forget that, and the adversarial trainers would not be able to basically detect and remove that. Yeah.
Okay, thank you.
Sorry, more question.
Oh, sorry.
Sure.
Quick question. Now I'm confused. Are you saying that you are doing a gradient reversal of the discriminator?
No. We're not doing a gradient reversal here. So we're basically, I mean, gradient reversal would be like taking the negative gradient and then like basically trying to minimize it. I mean, it could be also interpreted actually as a gradient reversal problem because you train a two-stage algorithm. So the first stage, you only maximize the weights of the encoder and the decoder. And the second stage, I mean, that's how you actually train GANs. Yeah, so you train the generator for some epochs, and then you actually train the discriminator afterward. Yeah, and then it's like this iterative back and forth defend police game. So we go back and forth. It's actually similar training plan here.
Helmholtz, I just want to ask you a quick question. Do you think for combinations, it's mostly predictions based on independent combinations of the drugs? Or do you think you can, I can imagine if you had enough perturbations, enough drugs, you can decompose something, learn something about, like pathways or synergistic drug effects possibly?
Yeah, I'll show you in a second, actually, that we can actually learn the synergies. But like the important part is that the model actually enforces this kind of like triangular relations, because like it learns that any combination should be a linear composition of like whatever single drugs that are there. But the important part is this linear composition is in the latent space. So it's not really linear in the gene expression. So the nonlinear effects could also be modeled as linear effects in the latent space. So yeah.
I see, but they're independent contributions fundamentally.
They're
right?
independent contributions. Yeah,
right?
Exactly.
So if you had synergistic effects in the latent space.
I mean, yeah, basically you can complicate this model and then have like interaction terms like A plus B and then AB for,
Interesting. Okay, I should probably just listen to you.
Yeah. Awesome. So I'll just show you one example.
I also have one question, Mohammed, if you go one step back. It's really great to see you, by the way. Yeah, I was just thinking about like how to balance the loss function. Like whenever I add like an additional term to the loss function, it becomes like a nightmare to just like think about how to balance it. So if you have like continuous things, continuous covariates and like discrete covariates, then you can use like a binary cross entropy and the mean squared error and so on. And then how exactly do you calculate the right level of loss for these individual terms? Are they like all hyperparameters, or like how are the users supposed to balance it compared to the like the
that's actually a good part about this, like Gökçen, that like we the only loss that we measure in this model is like the reconstruction loss between the predicted and real. And so that could be like if you have, depending on distribution, could be like a count or MSE. And the other loss is basically the cross entropy of the the perturbations. Yeah. And then if you have continuous variable on the perturbation level, then you can like simply convert the cross entropy into like KL loss. which then actually measure the distribution of the continuous variables for the discriminator. And it works quite well, actually, if you do that.
But if you have two categorical things, for example, one with 3 categories and one with 100, then if you, like for the adversarial loss terms, do they contribute like the same way, or do you have like additional tricks to make them like more balanced, but their contribution to the final loss a little like more balanced?
Oh yeah. So yeah, we have a penalty also for that. So we have a adversarial penalty for that, and then so the way that to balance this is like similar to GANs, like the tricks that you train GANs. So you either balance the number of iterations that you train generator here, the encoder decoder, and the discriminator. So that's one hyperparameter which is important. The second hyperparameter is the lambda and the discriminator loss, which is also a hyperparameter. And yeah, I mean, to be honest, there is not really a clear way to account for this. It's just like grid search. Yeah.
All right, I want to now interject and say that, Bo, you have, let's say, 6-7 minutes, to
okay.
Yeah. So guys, please, let's, if you have like, extended discussion, let's just sort of put it towards the end. Thank you so much.
Okay.
Just want to have good,
so I'll quickly go through some examples that we have. So we applied our model on I think this is the biggest perturbation data set at single cell level from Cole Trapno's lab so basically they were showcasing their cyplex technology, so multiplexing technology on 3 different cancer cell lines with 188 drugs and then 5 different dosages. And then we applied actually our model on this data set. So we had, close to 500,000 cells and then 5,000 different conditions. And this part is basically just to show that if you leave out part of the data, so basically this was a certain perturbation in MCF7 cells, and then you can see that the model actually can capture the top differentially and up and down regulated genes and upregulate or downregulate them. And then we again tested on different drugs, but I think this is not super interesting, just to showcase that it works well. The interesting part here is that we can model the continuous effects. So here you can actually perform counterfactual inference on like the dosages that you haven't seen, and the model basically can predict that a specific cell line respond, or a specific cell line basically respond differently according to different drugs. So it finds out, for example, this like aflatoxin drug is not, so this cell line is not responsive to that, which is in my opinion as important as predicting the response if you actually find out non-responders. And then on the interpretability, since we learned these embeddings, the model actually can learn embeddings for cell type or perturbations. And then it actually clustered similar drugs that responded to have like similar gene expression effects from this data set. So here you can see 3 major clusters. So basically epigenetic draw, cell cycle, and tyrosine kinase signaling. And it actually puts out drugs from these like mechanism of actions that has similar gene expression response. And again, you see this like in continuous space, you see that like the most responsive drugs, which actually corroborates what Cole had in their paper, that like basically these are the most responsive drugs for these cell lines. And then we validated this on applying our model on Link's data set and showed that basically similar drugs actually cluster together on a bulk level even, and similarly also for like cell lines and links. So one more interesting example would be that we applied it to predict the genetic knockouts, basically combinatorial genetic knockouts. Here there was a data set from Jonathan Weissman's lab, I think at MIT. And then we showed that again, instead of like having a perturbation space, we had like genetic latent space, and then showed that basically similar genetic knockouts which basically induce similar gene programs are grouped together in our gene latent space. And as you can see it here, so, and then we can predict like double knockouts. So this was like a CRISPR-I experiment. So basically we showed that like if the model actually hasn't seen a double knockout, it can predict a synergistic effect or epistatic masking effect, basically when A and B, when one of the interactions mask the other one. And but most interesting like application would be like to use this model for experimental design. So here, out of like, I think in this experiment, out of like 5,000 something double knockouts they could measure, obviously single cell experiment, they only measured like 144 of them. And then we predicted all of the missing combinations. And what you see here are not basically gene expressions; they're basically the embeddings of this, like the predicted knockouts. And then we had this, since we had this like embedding space, we could basically also report an uncertainty measure that measures that like most of the predicted cells that were not in the data, they actually they lie close to like the seen perturbations. And then we also have some clusters that with high uncertainty that are not in the training data. And then that requires further considerations when looking at them. Yeah. And then we showed that for one specific cluster, we looked at them and we found that like all the different knockouts that were not measured for the specific knockout, we can see different patterns. So you could have epistasis, dominant, synergy, like all different like kind of variations that you can have with this combination, even though the mall only has seen like a few of like KLF plus another genetic knockout, and the rest were basically predicted. And finally, I'll just skip this, but we show that the similar thing could be possible on a cross-species example. So basically on a cross-species example, you can like leave out part of the data which are from like different species. And then based on the other species, you can predict response for the species. And then we show that like the prediction actually captured a species of specific genes that are not present in the other example. And then obviously we also have failure cases. So I'll just skip that, but like, the important part is the model will basically fail on the events that it hasn't seen. So basically, if you have some neumorphic response, so a completely new response in genetic knockout, the model cannot predict that because that information was not available in the data set. So quick outlook. I'll quickly go up to so similar perturbation modeling could be performed on multimodal data. So you can have interversion and genetic knockouts, and then predict the response not on gene expression, but also on a spatial or chromatin accessibility or like other different levels. And then you can use this for experimental design. And then basically there are like cool applications for machine learning. You can use active learning here. And this is also quite closely related to domain generalization. And then uncertainty estimation is another area that one can work here. And causal inference, this is close, also related to causal inference. But I think most of the causal model would basically fail here. I just, when I was making the slides, I saw this paper from like last night in Europe's 2021. And it was interesting that basically the paper, it showed that latent variable models on causal latent variable models on like high dimensional real data set, they completely fail. And this was accepted in NURBS, but this, I mean, that shows that like some of the hype here is just like, yeah, it's basically hype. So we should actually take care and then consider these things. And then finally, another outlook would be just for the cases that, for example, for neumorphic or like the super auto distribution things that we don't see in the data, we can inject prior knowledge and hopefully predict those kind of effects. And then finally, like I thanks my collaborators, both from ICB, Fabian's lab, and then Facebook AI and other people. Thanks a lot.
Thank you, Mo. Actually, I have a quick question, but so please just spend like 1 minute to answer it, and then we could have it in the later discussion. So we had a fantastic speaker, I think 2 years ago, Anna Goldenberg, and she told us about a number of works that she had done. In particular, one of them that stuck with me is called like Dr. VAE. So that's basically like so their idea was to basically take cancer cell line data and then get patient-derived cell lines and basically see how much of your predictions on cancer cell lines can be sort of transferred to patient-derived cell lines. So one very interesting takeaway from that talk for me was that basically they said that we have a negative result, like in the sense that yes, we can do this with linear models, then we can do this with deep models. Deep models are like this much better than linear models, and also none of them actually quite work. So that was sort of my takeaway from that talk. Now, I guess my question here, I mean, I think this can be like a long discussion, is that with using deep autoencoders, is there a categorical improvement that we're seeing in our prediction capability? Or is it still, like a qualitative, like, small improvement over the linear model? Like, is it like all of a sudden we're learning that, okay, that cell type has this open chromatin, this has like this open chromatin, they're going to put 2 and 2 together, and now I'm going to decide something that a linear model could never do. Is it like one of those types of cases, or like you're still seeing like, just a quantitative improvement over linear models?
No, this is actually, we went deep into like this kind of question, because I mean, the first time, like not this paper, not the one before, but actually the first one that we did in the series of perturbation modeling SCGen, like 3 years ago, we actually compared to a PCA. What would happen if you do it with a PCA? And then the biggest take was like, like you can't do it with PCA, because PCA basically predict like response for all the cells, even though those cells would not, I mean, it's a linear model, so it cannot capture the nonlinearity. So anything which is like, which has nonlinear effects, we're like specific to a specific to a cell type and not present in the rest, linear models cannot capture that. Yeah. So I think that's I would say the biggest motivation to leave linear models, at least for this task, and then stick to like more complicated functions to model such things. Yeah.
Thank you. All right, let's continue that in the discussion. All right, David, please take it away.
Thanks. Now I'm muted, right? Yeah, perfect. Thanks. Cool. Yeah, thanks everybody. I'm gonna pick up on the point of latent space learning and representation learning that Fabian talked about earlier, and yeah, we'll just extend it with out of domain prediction. Specifically along the lines of independence assumptions and representation learning, and specifically IID assumptions. So IID. You might have heard this before, a term from applied statistics on the nature of the data generating process, if you want. It means that the data is independent and identically distributed. And this is something that's mathematically inherent in a ton of the models that we deal with that originally come from applied machine learning or applied statistics. For example, is inherent in embeddings that are learned per cell. So here on the right, show you a map of a sample, and in colors and clusters, we see different cell types, and then the structure within the cell type reflects within cell type variance and gene expression. So these are usually learned using loss functions that assume independence of data points, so cells. And similar also, like if you talk about differential expression analysis or other more low parametric statistical models, usually the loss functions assume that data points are IID. So why do we impose this? So first of all, the way we measure the system, especially throughout like the first years of single-cell biology, that usually was dissociating a tissue, essentially physically compartmentalizing a cell, and then measuring chemical properties of the cell. So there, the cell becomes an observation. I think this is like in a lot of ways like the origin of why people like treat cells as IID in a sample. However, if you think of the cell as coming from an organ, like for example, from your inflamed lung, is a cell in that lung really independent of the other cells in that lung? Biologically speaking, it's very much not so. So it's communicating with the other cells in that tissue, or at least in its proximity. It's subject to the same tissue level confounders. So let's say it's far away or close to blood vessel, and is therefore subject to oxygen or nutrient gradients. You could keep going on like this for like hours, I guess. There's essentially all of multicellular biology that confounds cells and results in cells not being independent of each other. It's the entire point of multicellular organisms, if you want. So, but what does that mean for us doing single-cell analyses? Maybe to answer that question, let's look at how this realizes in an analysis that assumes that the cells are IID. So let's go back to this embedding that we looked at. I circled two substates of cell type A and B. Let's assume that these tend to co-occur. So we either see these subclusters coming up in both cell types or not in either, if we look across patients. They could, for example, be because both are the inflamed states of that cell type, and that tissue is either inflamed, then I see the inflamed subcluster, or it's not inflamed, then I don't see it in either. This implies that we see the effect of this dependence between cells and the embedding, but having access to it would give us a stronger mechanistic handle on what's happening in the tissue. So now I want to go a bit into how we could go about identifying these dependencies. So there's a very popular stream in single-cell analyses right now that's centered around cell communication analyses. And I have a far from comprehensive list of tools and approaches here. A number of them are based on ligand and receptor gene expressions, for example, very popularly NicheNet and CellDB, but also tons of other approaches. Also approaches that take into account if genes downstream of receptor signaling are differentially regulated, which, for example, also NicheNet does. So these are approaches to use gene expression vectors of individual cells, sort of still treating them somewhat independently if you want, and look if there's like using biochemical prior knowledge, the possibility of these cells to interact, and then for us to form hypotheses on whether or not two clusters of cells might be interacting. Then in spatial omics, where we look at the position of cells and tissues, there's approaches on neighborhood enrichment. Again, sort of like people like looking at the potential of cell types to interact. If they're never close to each other, maybe it is unlikely that they ever interact. Now, looking back to the two talks before me, how would we deal with this in a representation learning setting? Right, so like all of the models that were discussed so far are essentially embedding a gene expression vector that is a cell into a latent space. What does it mean if there's dependencies between cells? Would I literally embed the entire sample into that latent space? So would I embed a batch times cell times gene matrix into a latent space? That seems not feasible in terms of like identifiability, because you can imagine we don't have so many samples and the weight matrices that are induced by this type of approach are gigantic. And probably also not very insightful, because you just like induce like a ton of parameters. But in principle, right, like if you think of dependencies to play a role in the setting, like this is something one might actually like naively start at. And in this talk, I want to talk about an alternative way of modeling dependencies between observations in cell-wise representation learning that uses space to break this problem down to a more reasonable level. And specifically, the way we use space in the set of models that I come to is as spatial graphs of single cells. And I want to spend one slide on explaining where they come from, what they are. So these spatial graphs of single cells are typically on an image structured data type that has subcellular resolution. So it could, for example, be RNA measurements in place or protein measurements in place. So imaging mass spectrometry, morphage, sequence codecs, methods that give you subcellular resolution of a tissue, of a 2D slice of a tissue, and then the channels are sort of gene expression of different genes. We can segment cells in that tissue. So here moving on the cartoon from the left to the middle, we sort of group pixels, so chemical pixels, into segments that are cells, and then based on gene expression similar to single cell workflows on dissociated data, we can assign cell types. And for example, we could like identify relations such as indicated here, that we have two cancer cells, an orange and red. One of them is next to a blue immune cell, and it is repressed because of the proximity to the immune cell, right? So this is an interaction that exists in tissues, and an interaction that could be intuitively analyzed given spatial relations of cells. So what we do with these segments, essentially directly harvesting this intuition, is to build a spatial proximity graph. So put a node for each segment and connect nodes that are within a spatial neighborhood, like at a particular radius or resolution. These neighborhoods, so here a cell and its spatial neighbors within like a given distance, these can be described as niches, for example, and niches are a really important like tissue biological level of abstraction towards emergence. So there's some properties of tissues that you can understand on the level of niches, which are really difficult to understand on the level of cells. So there's some really cool work from Gary Noel's lab, for example, about tumor infiltration by immune cells, and similar concepts are like really abundant in tissue biology. Now, to how we encode the dependencies between observations now in these spatial graphs. We do this in a framework that we framed as node-centric expression models. The idea is that every node on the graph is a cell, and we want to model the expression of the cell as a function of the graph. It starts with a very simple, essentially a linear model of this setting. So I want to predict the gene expression of a given node, given its cell type, and I assume that I can explain some of the within cell type variation of the cell as a function of the graph, or like the neighborhood in the graph. So here on the bottom, that means that if I embed the yellow immune cell into a gene expression space, I should get either, sorry, the yellow cancer cell, I should get either a repressed or a non-repressed cancer cell based on whether or not there's an immune cell in that neighborhood, right? So like I can frame this in principle as a linear model. That would be a model that can capture this dependency between those observations. Now, having fit such a model, I would then, if I framed this cleverly, be able to extract the interaction. So here, there's a directional interaction in this data set that goes from the immune cell to the cancer cell. So this is, interpretable in this way, and also like directly biologically tangible. Now, I think it's thinking about this in this linear model setting of binary, is a cell present in the neighborhood or not, and which cell type am I, is, I think it's a very nice intuition to like sort of like build on like how I arrive at a gene expression vector from cell types and graph, and how this relates to variance attribution, or how this relates to variance within a cell type. In a more general setting, we showed that this is an instance of a graph neural network, and in particular, a one-layer network with a very specific aggregator. But in principle, if you think about this task of having a graph of cell types in space that come together to form specific cell type specific interactions that then yield very specific expression states, so very specific gene expression vectors. This is a very general graph representation learning problem where I go from a coarse feature space in the input to a very fine-grained feature space in the output. And you could think about adding more layers to adding higher, so that means adding higher abstraction through more parameters in depth in the model. But you could also think about using different graph or like local niche aggregators here. So linear convolutions over the graph, or like graph attention from graph attention networks, max poolings, you name it, essentially. So we benchmarked this class of model on a couple of data sets. And first we wanted to see if we can capture any dependency of cell expression states on the neighborhood at all. And to do that, what we did is essentially to fit models that try to explain gene expression as a function of cell type without having access to the space. So here on the on the x-axis, that is the size of the neighborhood that's always the 0. So that's a baseline non-spatial model. And then as I move to the right for each of these 5 data sets, I increased the neighborhood up to a point where the neighborhood is so large that it's essentially non-informative. And then on the y-axis, I show the predictive power of the network. And so what we see is that for each of these 5 data sets, this model has a predictive peak at an intermediate length scale that is a bit larger than an individual cell's radius. So this is biologically the type of dependency that you would expect if you have between cell communication or between cells, statistical dependency, right? Like a bit larger than a cell, like somewhere they're still like in vicinity. So there's some, it's reasonable to assume that there's molecular communication going on. Yeah. And then the performance dropping at the end is sort of like our control that as this local niche becomes uninformative because it's essentially the entire image, like the performance also drops. So this shows that this type of model can actually capture these dependencies and a representation in context. Now, having shown that, we can look into a lot of interesting properties of these models because they're obviously very expressive about which cells interact with which cells, like what cell types they tend to be, what genes are involved, right? There's like arbitrary amount of interpretation you can do. So the first one would be looking at the length scale, so the dependency of the predictive power for individual signal receiving cells, so predicted cells. And even there already, you see like really strong differences. For example, if you look at the first two B cells and endothelial cells, so the predictive power peaks at like really different length scales. And in principle there, right, like different tissues, different cell types could be communicating via drastically different ways with their environments and would therefore also have drastically like different niches. So these are things that we can describe with that model. Next, you have an example of mouse cortex measured with Murfish. On the left panel, there's one cell type that we subclustered. And then in the middle panel, cell type in its vicinity. And we sort of see that essentially the niche structure here sort of like follows a layered structure. This entire organ is layered. And therefore, like also like the niches are somewhat layered. And you would expect that if you're good at predicting particular niches, you'd see that sort of like as a layered predictive advantage. And this is also here on the right, predictive advantage over a baseline model. This is what we see. Like there's like a couple of niches that we are good at predicting. Yeah. Then we can attribute this to subclusters. So we could look at these subclusters and specifically look in the niches where, so what cell types are present in this niche, which are like the putative center cell types. We can look at individual genes. So we can, in the linear model setting, through differential expression testing, essentially testing like parameters of this model in a deeper network, maybe via saliency, look at, for a given gene in a signal or dependency receiving cell, which cell type in the neighborhood does this look at, like what cell type does this depend on? So for example, here's CD8 T cells in this particular inflamed colon sample tend to look at PDL1 plus cells that carry the ligand, and the gene that seems to be involved is P1. Yeah, so this, yeah, because you can break it down to genes that easily, it's then really nice for hypothesis building. Now, on the last part, extending on the latent state modeling. So what I showed you now is essentially supervision, right? It's graph representation learning, but it's essentially a problem that is supervised on each node. We could also model node-wise latent states. For this, we turn this supervised network into one that has variational states in it, essentially turning this into a conditional variational autoencoder where we push through cells and we embed the graph as a condition. Yeah. So we call this node generative modeling that could have the advantage that you can capture like other modes and confounding latent states like cell cycle and stuff. We use that to transfer cells into unseen neighborhoods. So for a given cell type, try to predict how that would look like in a new neighborhood. So this is a style transfer task if you want to an unseen neighborhood. So like neighborhood transfer or something like this. The task as in general for style transfer depends, or conditions for this conditional type of modeling, depends on the model to attribute as much signal as possible to the condition rather than to the latent state. And this is pretty difficult in this type of setting because of the really complex condition that is graph embedding. So they're essentially, so far we have the negative side. We played a bit around with approaches to create like stronger posteriors that maybe allow us to reflect this really complex condition better. So yeah, throwing in an idea that we tried, for example, is on lagging encoder networks. So there's a bunch of machine learning literature on that. Yeah, I'd like to finish on a couple of outlooks. So this last point that I made, this type of out-of-domain prediction is pretty hard, and it may be that there's like some algorithmic work to do there to like really improve models that have graph embeddings as a condition, and also like propagate gradients through them. Technologically, you see like really strong advances in the spatial omics technologies right now. So the resolution of the cell states that we model and the abstraction that we can reach, like, is really increasing. So that might allow us to do a lot cooler things in a year from now than we're doing now. And then lastly, on the tissue biological assumptions that go into this and the type of phenomena that we model. So this, because of the snapshot at the time of the spatial omics, essentially assumes that these cells live in these stable niches, right? But that it doesn't hold true for all tissues or for all cells in every tissue. There may be like transient cells like migrating through tissues. So there is like some interesting extra stuff to potentially consider in the future. So looking back to the ID discussion at the beginning, and also representation learning in general. So when could it make sense to drop this independence assumption? Probably if you look at tissues, right? So because all of this that I talked about now is like, what is the niche, what is the tissue? It's like really a different view of this type of biological system. Yeah, and then lastly, I would like to thank especially Anna Shah, who's co-first on this author, and Fabian, my co-authors, and then also Sabrina, Stefan, Christian, Sebastian, Dirk, Xiao, and Meng for talking to us about this project. Thank you.